Okay. Good morning, good afternoon, everybody. Um, I am homesick. I have a very bad sore throat, and I didn't really feel like yelling and talking all day and making it worse. So, I'm doing a video demonstration of the lab. I know this is not the most fun thing in the world, but I do promise end of this week we are doing a lab and you will get to eat food with the lab. So, look forward to that. Okay? But for right now, you should be on page 16. Oh, I don't know if it's going to show you the pages. Hold it in front of me. Okay, page 16 has this drawing on it. Other side. I'm looking at it backwards. Okay. And at the top it has the lesson 3 essential question. Why do molecules in the small intestine seem like they are disappearing? Okay. So we are going to do a lab to sort of take a closer look at that. Unfortunately, we can't get an actual small intestine in here. We don't have the funds. Okay. So, what I am going to have us do is we are going to use something called dialysis tubing. It works very similarly to the way a small intestine works. Okay. So, look at this diagram. Okay. It is on the bottom of your page 16. Okay. And it says label the diagram below. What does the dialysis tube represent? What does the liquid outside of the dialysis tube represent? And where should we place the food molecules in our model? Okay. And why are we placing them there? Okay. So the dialysis tube is going right here. That is going to represent our small intestine. Okay. So on your thing, I'm writing very badly. I'm sorry. Okay. This is our small intestine. Okay. And then the water on the outside, that's going to represent our bloodstream. Okay. So dialysis tube is our small intestine. The water on the outside is our bloodstream. When we eat food, our food molecules are going to run in through our small intestine. So we want to put our food molecules inside of our small intestine. Okay. So that is our food. Okay. So make sure you have all of those labels on your diagram on page 16. Okay. And when you have all of that done, then you are going to flip over to page 17. At the top, it says dialysis tube investigation. Okay. Clear all drawings. Okay. So we know that the dialysis tube is going to represent our bloodstream. So I have the dialysis tube set up here. Okay. I have one side tied off with some dental floss. And on the inside, we're going to put some food. Okay. Inside, we are going to place, we're going to use a dropper. And inside it is a mixture of water and some smaller sugar molecules. Okay, the smaller sugar molecules are whatchamacallit? Why is my brain blanking today? The smaller sugar molecules are glucose and some larger sugar molecules, starch. Okay, so we are going to place that inside of the tube and we're going to work on filling it up. Okay. So now we have the food molecules inside of the tube. Okay. And we're going to fold it back over and we're going to tie it. Tie it, knot it a couple times, and of course my lights turned out. Let me grab that. Okay. So now we have our dialysis tube, okay? And it's filled with the food molecules and water. And we are going to place it into just a cup of water, okay? And we would leave it sort of suspended, so we'll tape the sides, okay? But then it's going to be hanging out inside of the tube, okay? And we're going to leave this to sit overnight. I actually set up this on Friday so we don't have to wait, and I already have the results, okay? So we're going to use something called an indicator, okay? Let me back up so that way you actually have the correct information. So it says part 1, how do we set up our dialysis tube system? Which food molecules are outside of the tube to start? Okay, there you want to write none. Okay, there are no food molecules outside of the tube to start. Which food molecules are inside of the tube to start? That is going to be glucose and starch. None. And then we have glucose and starch. Let me get it so you can actually see my screen. Okay. So we have none, and then glucose and starch are on the inside. Okay. Next, predictions. Which food molecules do you think will be found outside of the tube at the end? And which food molecules do you think you'll find inside of the tube at the end? Okay, so I want you to write down in those boxes. Go ahead and pause here. Okay, under the outside, you can either write glucose, starch, or none. Okay, you could write glucose and starch. You could write just glucose, just starch, or none. Okay, and then under inside, you can write just glucose, you could write just starch, or you could write none. Okay. And then there's an empty space in the middle. I want you to write down why you made the predictions that you made. If you need to pause the video here, so that way you can write down your predictions, you can go ahead and do that. Okay. Now we're looking at part 3. It says, what do iodine and Benedict's look like with just water? So iodine and Benedict's are both indicators, meaning that they tell you if something is present. Okay. Iodine is going to indicate whether starch is present. Okay, it comes in a dark glass jar like this. Okay, and when it is just in water, it looks like this. Okay, so write down what color you see when iodine is just in water. Okay, Benedict's is going to tell us if there is glucose present. So sugar, tinier sugar molecules. Okay, and this is the color of Benedict's just with water. Okay, so write down the color. That is in part two. Okay, you can see the picture here. Color of iodine in water, color of Benedict's in water. Okay. Next, okay, when starch is present, okay, iodine is going to become like a dark brown or a black color. Okay. When glucose is present, Benedict's will change color from like a blue to a red. So what we're going to do is we are going to take a sample from what is outside of the tube, okay? And we are going to place it into these tubes. This one's halfway filled with iodine, this one's halfway filled with Benedict. And then we're going to pour some hot water over it, and we're going to look at it after the change. I'm going to heat up my water a little bit more. Okay. We are looking at just what is outside of the tube. Oh, I grab the cup. Okay. I filled up the tiny tube with the outside liquid and Benedict's, and I'm going to put it into the styrofoam cup. And then I am going to fill up the tiny tube of iodine with the liquid outside. Okay, I'm going to put it in the cup. Then I'm going to pour some hot water over the two of them, and that's going to sit for a little bit. Okay? In the meantime, what we can do is we can look at part 5 while we wait for that to sit, and we're going to look at what is inside of the tube. Okay? So notice the tube still has a lot of liquid in it. Okay? And I'm going to carefully cut it open and take the liquid that is inside the tube and also put it into these tubes. Okay, first the container with iodine. And next, the container with Benedict's. Okay, we'll seal these and pour some hot water over them. Okay, we're giving some time for these to settle. Hmm. Okay, so we should have part 3 done where color of iodine in water is brown, color of iod or Benedict's in water is blue. Okay, now we are going to look at the food molecules that are outside of the tube after the investigation. Okay, we look at the outside. Our Benedict or our iodine. Yeah. The outside, our iodine is still that reddish brown color. Okay, so it has not changed. So color of outside liquid with iodine, we're going to write brown. Okay, you could write light brown, reddish brown. Okay, but it's still a brown color. Now, I didn't give this the full time to process. So I'll show you a color comparison here. But this is the color of our Benedicts, okay? And this is just Benedict's in water. Okay, so you can see how this one has gotten like pale. Okay, and it's turning almost green. Okay, so color of outside liquid with Benedict's, I want you to write greenish blue. I'm going to give a shout out to Shika because I hear her talking in the hallway right now. Okay. And then, so food molecules detected outside of the tube, we are going to write glucose. Okay. Those are the food molecules that have been detected outside of the tube. Okay. Now, on part 5, okay, on page 18, we are going to look at the color of the iodine from inside of the tube. Okay. And you're going to see that this is a really dark brown, almost black. Okay. So you can write down dark brown. And then the Benedict's, again, is that darker greenish grayish blue. So that means the food molecules inside of the tube are going to be starch and glucose. So this is what you should have. Okay. Now there are the sense-making questions. Okay, part 6 says, did any of the food molecules that started off inside of the dialysis tube end up outside of the tube? Explain what happened. Number 2, what do the class results tell us about the structure of the surface of the dialysis tubing? And number 3, look at the structure of a complex carbohydrate molecule like starch compared to a sugar molecule like glucose. What do you notice and wonder? And there are pictures of a glucose molecule and part of a starch molecule. Okay. So I want you to answer those 3 questions. Okay. And then that will be the last thing for you to do today. Tomorrow, we will be writing a CER. Okay, claim evidence reasoning about what happened. And we'll be taking some notes on cell transport. Okay. Depending on timing, we might even start the gummy bear lab tomorrow. So, if you have any questions on this, I will be back on Wednesday and we will discuss it. Okay. Trying to think if there's anything else I wanted to say. Okay. So by the end of class, pages 16 through 18 should be completely done. They go into your binder and you put your binder back over on the counter. Okay. Don't turn it in. But today you did your do now, page 16, page 17, and page 18. Okay. I hope you have a good day. Please behave for whoever the sub is. Love you. Bye.